In today's video, I'm going to be covering the topic of eukaryotic cells and organelles. Eukaryotic cells are one of two main cell types that we cover in A-level biology, with the other cell type being prokaryotic cells, which I will cover in a separate video. But just to note some of the main differences between the two. Prokaryotic organisms are single-celled organisms, and they are made up of a singular prokaryotic cell. Prokaryotic cells are simpler cell types and they're smaller than eukaryotic cells. So let's look at a bacterium, for example. Bacteria are a much simpler cell type. They contain less organelles and they're much smaller, and one bacterium consists of one prokaryotic cell. Eukaryotic organisms, on the other hand, are made up of eukaryotic cells. The cell type is more complicated, and they can exist as animal cells, plant cells, algal cells, and fungal cells. And eukaryotic organisms are, for the most part, multicellular organisms. The only example I can think off the top of my head is an amoeba, which is a single-celled organism. Both prokaryotic and eukaryotic cells contain organelles, which are cell components, and they each have a specific function. So you can see in the centre of my screen now that we have a labelled animal cell. These are often represented as a sphere with a cutout so you can see the structures or the organelles that exist inside of the cell. Now, some of these you should be familiar with from GCSE biology. So structures such as the cell membrane, the ribosomes, the nucleus, the mitochondria, and the cytoplasm should already be very familiar to you. In A-level biology, we look in more detail at some new structures for you. So these include the rough and the smooth endoplasmic reticulum, the Golgi apparatus, and the secretory vesicles, as well as several others, which we'll go into more detail later in this video. But it is really important that you are able to recognise the structures that exist inside of both animals and plant cells, because you could potentially get questions where you have to label some of these structures. Now let's move on and have a look at a plant cell. Again, this should be a diagram that you are very familiar with from GCSE biology. We can see that a lot of the structures within the plant cell are the same as in animal cells, but there are obviously some differences, and that is because plant cells have a very different function to animal cells. So plant cells have many of the same organelles as animal cells, for example, mitochondria, ribosomes, smooth endoplasmic reticulum, and a nucleus, but they have several extras as well. This includes the cell wall, which is made up of cellulose, which is a type of sugar, and these contain something called plasmodesmata, and these are channels that allow substances to be exchanged with adjacent cells. So essentially these plasmodesmata are almost like little doorways, so that substances from one plant cell can travel through into another plant cell. Plant cells also contain a vacuole, which is a compartment containing cell sap, and it can also be used to trap in toxins. They also contain chloroplasts, which you should know from GCSE, help a plant to photosynthesize and produce its own sugars. Plant cells also contain something called starch grains, and these contain excess plant sugars, but these aren't actually considered as organelles. So the last cell type that we won't cover in very much detail are algal and fungal cells. You don't need to know about these cell types in detail. However, you may be asked to label certain structures in an algal cell, for example, like I've got on screen, and you may also be asked about how its structure relates to its function. So if we have a look at this algal cell on screen, we can see that it's got two flagella, and if we think back to learning about bacteria in GCSE biology, we know that the flagellum allows the cell to move and almost swim along. So you could, for example, to answer that question, say that the flagella allow it to be motile and move. It also contains a cell wall, which obviously, because algae is found in watery substances, this helps to prevent water coming into the cell, so it protects it from bursting by osmosis. And we can also see that it contains many chloroplasts, which is obviously very useful for photosynthesis. So the primary difference between algal and fungal cells. The algal cells are very similar to plant cells, sharing many of the same organelles, so chloroplasts and cell wall. The only difference I can see from this diagram is the flagella that algal cells contain. Algae, like plants, also carry out photosynthesis, hence the many chloroplasts that we can see in the cell. And algal cells can actually be either single-celled organisms or multicellular organisms. Fungal cells, on the other hand, so for example mushrooms and yeast are two very good examples, are similar to plant cells, but their cell walls are made up of chitin instead of cellulose. And fungal cells don't contain chloroplasts as they don't need to photosynthesize. Actually, many fungi use their roots and they form what's called a symbiotic relationship with other surrounding plants. But that is a topic that will be covered in a much later video. So let's now have a look at some of the organelles that you would find in animal and plant cells and what their function is. So the first one is the plasma membrane. And this is the membrane that is found on the surfaces of animal cells and on the inside of the cell wall in cell types that contain a cell wall. So it'll be the cell wall, and then directly next to it is the plasma membrane. The plasma membrane regulates movements of substances in and out of the cells. And again, this is something we will cover in a later video, the process of how various substances are able to be transported in and out of cells. They also contain receptor molecules, and this allows it to respond to various chemicals, such as hormones. Next one is the nucleus. You can see the diagram on screen. The nucleus is quite a complex structure and has many supporting structures that allow it to function properly. So the nucleus is a large organelle surrounded by a nuclear envelope, which has a double membrane, and pores, which allow various substances to be transported through it. The nucleus contains chromosomes, and chromosomes are made up of protein-bound linear DNA, and it also contains smaller structures called a nucleolus, which is involved in making ribosomes. The nucleus controls cellular activities such as transcription of DNA, and it gives the instructions to make proteins, so it can almost be considered like the brain of the cell. It controls everything that's going on within the cell. And the pores in the nucleus, these nuclear pores that you can see labelled on the diagram, allow substances to move between the nucleus and the cytoplasm. Our next structure is the mitochondria. This is an oval-shaped organelle, and it also contains a double membrane, like the nucleus, and the inner membrane is folded to form a structure called cristae. Inside the mitochondria, we have the mitochondrial matrix, and this contains enzymes involved in respiration. And mitochondria is the site of aerobic respiration, where ATP is produced. They're often found in large numbers in cells, but especially in very active cells that require lots of energy. Our next structure is the chloroplast, and these are small flattened structures that are found in plant and algal cells, and they also have a double membrane. They have internal structures called thylakoids, and these are surrounded by membranes, and they are stacked on top of one another to form a structure called grana. These grana are then linked together by something called a lamellae, and this is a thin, flat section of thylakoid membrane. Chloroplasts are the site of photosynthesis, and some photosynthesis occurs in the grana and some in the stroma, and the stroma is a thick fluid that's found in the chloroplasts. Next one is the Golgi apparatus. You can see this odd green looking structure on the screen. It almost looks a bit like a pyramid. And the Golgi apparatus is a group of flattened, membrane-bound, fluid-filled sacs. New lipids and proteins are processed and packaged within the Golgi apparatus, and it sometimes can synthesize lysosomes. Next is the Golgi vesicle. Again, we have the same diagram here. The vesicles, however, are these small little bubbles, these yellow and green circles that you can see on the diagram. And these are small fluid-filled sacs in the cell cytoplasm, and they're surrounded by a membrane produced by the Golgi apparatus. Their function is to store lipids and proteins that are made by the Golgi apparatus, and then they transport these substances out of the cell via the cell surface membrane. Our next structure is the lysosome, which we've mentioned. They can be synthesized by the Golgi apparatus. And a lysosome is a type of Golgi vesicle. They're round organelles, and they're surrounded by a membrane, and they have an internal structure that's a mix of hydrolytic enzymes. Their actual internal structure is largely unknown to us, so this diagram may not actually be accurate as to what they look like inside. They contain digestive enzymes called lysozymes, and these are kept in the centre of the cell, and they are kept separate, these lysozymes, from the cytoplasm by surrounding membranes, and their function is to digest invading cells or break down worn cell components. So when we cover the immune system, we will see the function of lysosomes and lysozymes much more in that topic. Our next structure is the ribosome, something we should all be quite familiar with by now. And the ribosome is the site of protein synthesis within a cell. They are a small organelle, and they either freely float in the cytoplasm or they're attached to rough endoplasmic reticulum. They're not surrounded by a membrane, and they're made up of RNA and proteins. And again, we will have a look at ribosomes in much more detail, but they also contain this large subunit and a small subunit. Next is the rough endoplasmic reticulum, which is also shortened to RER. And you can see it's this very strange looking, it almost looks, these flattened line-like structures that you can see in the diagram, they almost look similar to the cristae that you see in mitochondria. And the difference with rough endoplasmic reticulum and smooth endoplasmic reticulum is that rough endoplasmic reticulum has ribosomes attached to it, which give it this bumpy appearance. Perhaps explains the name rough endoplasmic reticulum. And RER is a system of membranes that enclose a fluid-filled space, and the surface of them, as I've already said, is covered with ribosomes. And what its function is to fold and process proteins that have been made by these attached ribosomes. So if we look at smooth endoplasmic reticulum, we can see the structure is kind of similar to the rough endoplasmic reticulum. We can see that it's just consists of membranes though, and a fluid-filled space, doesn't have any attached ribosomes. So as I've already said, it's similar to the rough endoplasmic reticulum and has no attached ribosomes, and its function is to synthesize and process lipids. The cell wall is our next component, and you can see it's this hexagonal looking green structure, and the cell wall is a very rigid structure, and it surrounds plant, algal and fungal cells. We already said that in plant cells the cell wall is made up of cellulose, and in fungal cells it's made up of chitin. And its function is to support cells and prevent changes in the shape of the cells. Our final structure is the cell vacuole, and this is a membrane-bound organelle found in the plant cell cytoplasm, and its surrounding membrane is known as a tonoplast. The vacuole contains cell sap, and this is a weak solution of sugars and salts. And its function is to maintain internal cell pressure, and it keeps the cell rigid to stop plants wilting. And it also isolates any unwanted chemicals within the cell. So cells can specialise and contain various organelles which allow them to carry out a particular function. So in multicellular organisms, cells have to specialise to carry out specific functions. And so cell specialisation is what allows your brain to be very different from perhaps your stomach or your skin. And cell structure helps the cells to carry out this function. So the examples I've given here, cells that synthesize proteins will clearly need a lot of ribosomes, and cells that need lots of energy will need lots of mitochondria. So on the right hand side of the screen here, I've got a diagram of the lumen of the small intestine. Now, the lumen of the small intestine is made up of epithelial cells, and these form structures called villi, which are these large dome-like structures that you can see. And on the very right-hand side of the screen, the enterocyte cells that make up the villi are individual cells that then have these even smaller structures on, called microvilli. And the villi and the microvilli serve to increase the surface area of the cells, meaning that there's more surface area for food absorption. And they also contain many mitochondria, which is not shown in the diagram, but this increases the energy available to transport digested food. So in complex multicellular organisms, such as humans, specialised cells will develop, and these will group together to form a tissue. So for example, the skin is a type of tissue, but the cells in the epidermis and the dermis and the hypodermis will all be different from each other. So epidermal cells are a specialised cell type, and they function together with the dermal and hypodermal cells to form the skin tissue. So a tissue is a group of cells that work together to perform a certain function. So in the case of the skin is there to protect you from infection, and it's there as almost a barrier against water. So different tissues will work together to form an organ, and different organs will make up an organ system. So the example I've given down here, epithelial cells, various different epithelial cells will make epithelial tissue. Epithelial tissue combined with muscular and glandular tissue will form the stomach. And then the stomach with the liver, the small intestine and the large intestine will form the digestive system. So let's recap what we've covered today. We've learned that prokaryotes are single-celled organisms that are made up of a singular prokaryotic cell, and this will be covered in much more detail in my next video. We've learned that eukaryotic cells make up much more complex organisms such as animals, plants, algae and fungi. We know that plant, algal and fungal cells contain a cell wall. We know that organelles have a specific function within the cell, and that cells can specialise so that their structure and function aids with a particular task and contributes to the formation of tissues, organs and an organ system, and that cells that do specialise will contain organelles that will aid with their function. I have not included in the recap the structure and the function of the various organelles, because that will essentially just be repeating what I've told you in this video. But the best thing to do with that, especially with the newer structures, is just to go back every so often, go over their structure, what they look like and their function, because there are a lot of them, but eventually with continued practice it will sink in and you will remember all of them eventually. So the best thing to do now is to go and recap the content of this video and then practice the following test questions. Number 1, name 3 types of organelles in plant cells that are not found in animal cells and explain their structure and function. Number 2, from production to secretion, order and list the organelles that are involved in synthesizing hormones. Number 3, explain how intestinal epithelial cells are specialized for their function and how they are involved in making up the stomach organ and the digestive system. And number 4, plasma cells are specialized to produce antibodies, which are a type of protein. What organelles would be abundant in plasma cells and how does this help with their function? That's all for today. Thank you for listening.